Good afternoon and welcome back to AP Biology Semester 2. We are still in Big Idea, Free Energy, and we are in the 7th chapter today, Section 4, talking about active transport using energy to move solutes against their gradients. Despite the help of transport proteins, facilitated diffusion is considered passive transport because the solute is moving down its concentration gradient. Facilitated diffusion speeds transport of a solute by providing efficient passage through the membrane, but it does not alter the direction of transport. Some transport proteins, however, can move solutes against their concentration gradients across the plasma membrane from the side where they are less concentrated, whether inside or outside, to the side where they are more concentrated. Okay, looking at this, we find the need for energy in active transport. To pump a solute across a membrane against its gradient requires work. The cell must expend energy. Therefore, this type of membrane traffic is called active transport. The transport proteins that move solutes against a concentration gradient are all carrier proteins rather than channel proteins. This makes sense because when channel proteins are open, they merely allow solutes to flow down their concentration gradient rather than picking them up and transporting them against their gradient. Active transport enables a cell to maintain internal concentrations of small solutes that differ from concentrations in its environment. For example, compared with its surroundings, an animal cell has a much larger concentration of potassium ions and a much lower concentration of sodium ions. The plasma membrane helps maintain these steep gradients by pumping sodium out of the cell and potassium into the cell. As in other types of cellular work, ATP supplies the energy for most active transport. One way ATP can power active transport is by transferring its terminal phosphate group directly to the transport protein. This can induce the protein to change its shape in a manner that translocates a solute bound to the protein across the membrane. One transport system that works this way is the sodium-potassium pump, which exchanges sodium for potassium across the plasma membrane of animal cells. Okay, now how do ions pump maintain membrane potential? So glad you asked. Well, although all cells have voltages across their plasma membranes, voltage is electrical potential energy, a separation of opposite charges. The cytoplasm is negative in charge relative to the extracellular fluid because of an unequal distribution of anions and cations on opposite sides of the membrane. The voltage across a membrane, called a membrane potential, ranges from about 50 to 200 millivolts. The minus sign indicates that the inside of the cell is negative relative to the outside. So it would be written as negative 50 to negative 200 millivolts. Okay? The membrane potential acts like a battery, an energy source that affects the traffic of all charged substances across the membrane. Because the inside of the cell is negative compared with the outside, the membrane potential favors the passive transport of cations into the cell and anions out of the cell. Thus, two forces drive the diffusion of ions across a membrane: a chemical force, the ion's concentration gradient, and an electrical force, the effect of the membrane potential on the ion's movement. This combination of forces acting on an ion is called the electrochemical gradient. In the case of ions, then, we must refine our concept of passive transport. An ion diffuses not simply down its concentration gradient, but more exactly down its electrochemical gradient. For example, the concentration of sodium ions inside a resting nerve cell is much lower than outside it. When the cell stimulated, gated channels open that facilitate sodium diffusion. Sodium ions then fall down their electrochemical gradient, driven by the concentration gradient of sodium and by the attraction of these cations to the negative side of the membrane. In this example, both electrical and chemical contributions to the electrochemical gradient act in the same direction across the membrane. But this is not always so. In cases where electrical forces due to the membrane potential oppose the simple diffusion of an ion down its concentration gradient, active transport may be necessary. All right. Some membrane proteins that actively transport ions contribute to the membrane potential. With each crank, let me see. Let me talk about that right now. How about, let's talk about this, okay. A transport protein that generates voltage across a membrane is called an electrogenic pump. The sodium-potassium pump seems to be the major electrogenic pump of animals. The main electrogenic pump of plants, fungi, and bacteria is a proton pump, which actively transports hydrogen ions, protons, out of the cell. The pumping of hydrogen transfers positive charge from the cytoplasm to the extracellular solution. By generating voltage across membranes, electrogenic pumps store energy that can be tapped for cellular work. One important use of proton gradients in the cell is for ATP synthesis during cellular respiration, as you will see in Chapter 9. Another is a type of membrane traffic called cold transport, as I have both on the board, as you can see. Let's talk about cold transport for a minute, and how it's coupled transport by a membrane protein. Okay, so a single ATP powered pump that transports a specific solute can indirectly drive the active transport of several other solutes in a mechanism called cold transport. A substance that has been pumped across a membrane can do work as it moves back across the membrane by diffusion, analogous to water that has been pumped uphill and performs work as it flows back down. Another transport protein, a co-transporter, separate from the pump, can couple the downhill diffusion of this substance to the uphill transport of a second substance against its own concentration gradient. For example, a plant cell uses a gradient of hydrogen ions generated by its proton pumps to drive the active transport of amino acids, sugars, and several other nutrients into the cell. One transport protein couples the return of hydrogen ions to the transport of sucrose into the cell. This protein can translocate sucrose into the cell against a concentration gradient, but only if the sucrose molecule travels in the company of a hydrogen ion. The hydrogen ion uses the transport protein as an avenue to diffuse down the electrochemical gradient maintained by the proton pump. Plants use sucrose and hydrogen ion co-transport to load sucrose produced by photosynthesis into cells in the veins of leaves. The vascular tissue of the plant can then distribute the sugar to non-photosynthetic organs such as roots. What we know about the co-transport proteins, osmosis, and water balance in animal cells has helped us find more effective treatments for the dehydration resulting from diarrhea, a serious problem in developing countries where intestinal parasites are prevalent. Patients are given a solution to drink containing a high transport protein. Patients are given a solution to drink containing a high concentration of glucose and salt. The solutes are taken up by transport proteins on the surface of intestinal cells and pass through the cells into the blood. The increase in the blood solute concentration causes a flow of water from the intestine through the intestinal cells into the blood, rehydrating the patient. Because of the transport proteins involved, both glucose and sodium ions because of the transport proteins involved, both glucose and sodium ions from salt must be present. This is why athletes consume solute-rich sports drinks. And we are going to stop here for today and pick up on 7.5, talking about the bulk transport across the plasma membrane occurring by exocytosis and endocytosis. So, until next time, ta-ta!